Yeah. Yeah, I'm going to do that. And I'll talk about that first and just want to show you. Okay, can somebody confirm that you guys can hear me?
Yes.
I can hear you.
Is there a slight echo?
No.
You didn't
No,
hear
it
that.
sounds
Okay, I'm
fine.
hearing a slight echo. I'm not sure what that is. All right, let me start by saying I've done a fair amount of grading. So you should take a look at that. I got labs 00 and lab 01 graded, as well as the first lecture worksheet. And I got something else graded. The extra credit study worksheet, meaning where you put in the times where you're studying. That's not really graded. It's just if you submitted everything correctly, you got 6 points. If you submitted only your study times, then you got 3 points. Any question about any of that? Let me see if there's anything else I want to talk about the grading. The Plagy quiz has been graded. I don't do that. It's system graded itself. Sophia, if you're on, you did not get 100% on the Plagy quiz. Let's see if there's anything else I need to talk about that. Worksheet lecture 1 was graded. If you do not know the answer to the question and you don't want to look it up, don't answer it because you will not get a second chance in the lecture worksheets. You should wait until you get a question that you can answer. And do not skip the questions. I had one student skip the question. I don't know why they did that. It just makes it hard for the grading if you do not answer the questions in the order. If you don't like the current question, just don't answer a question and come back to it later, because that question will be answered by other students. And then you can answer a question later in the worksheet. I think that's it about the grading. You do have until this Saturday, 11:59 p.m. to resubmit answers to lab 00 and lab 01. Yeah, there is one other thing I need to mention, and that is this class in particular is writing comments in the comment section of the lesson. We'll just put it the lesson. And if you want to add a comment, put it in your document that you're submitting for the lesson. You can either put the comment at the top if you want me to read it first thing, or you can put it at the bottom. I will promise it will get read. Do not use the comment section in the grading. Let me see if I can find one. Don't really want to show a student's one, but let's just go to one. And I better shut down the grade book. Guess I've already shut it down. All right. If there aren't any submissions, it looks like I can't show you the speed grader. Oh, this is a lecture. That's why that won't work. Let's just go to another one. What I should do is go to a previous term and I could show you something. Oh, let's just go to that. That should work. All right. That's fine. Scroll down here. And I'll show you now on my screen. So if you want to add a comment, put it directly in your document. That would be here. You can put it at the top or at the bottom of the document. Do not put it in the assignment comments. This section is for me, and I will tell you in the lab, the errors for the first grading or what you got wrong on the second grading of the lab. And it'll be a place where I'll put comments about your assignment. If this is very large, it makes it difficult to work with. If you make a large comment in here, I promise I will delete it because it just interferes with my work. So please don't use the assignment comments, use it, any comment, put it in your direct document, and then submit it in the normal session. I don't have a download button because I'm the instructor. See if I can even back up to that. But just add it to your document. I can't even back up to that. All right. Any question about any of that?
But you are recording, right?
I should be. Let me stop the screen. Take a look. Yeah, I'm recording. Like I said, I can't see that really well unless I stop sharing. All right. Any questions about anything before we begin?
I had a question about how quickly you get back to us through Canvas emails. I know you mentioned it the other day, but I can't remember.
There were a lot of Canvas emails telling me about what the infectious disease project you guys wanted to do. And I wasn't going to start that until today, but I decided to just keep those emails unread, and I will read them after the lesson today, and then we'll get back to you about that. So if it is in regard to an infectious disease project, that has been put on hold because that project isn't beginning until today. Okay?
Okay,
So
thank
is that
you.
what it's about? All right. But I decided this term I wouldn't write you back and say you have to wait until Tuesday to begin that project. I just decided to hold the emails unread, and I'll get to you on that. All right, so what we're doing today is January 10th, and we are going to be continuing chapter 1 and hopefully starting chapter 2. I don't know if we'll get into that. Let me warn you, you do have the practice quiz 00, which is due, I believe, at 8 p.m. Wednesday. Let me look that up. Hopefully that's not going to be the grade book. There are two parts to the quiz. Quiz 00A is going to give you a fill in the blank or short answer question. And then quiz 00B will give you a multiple choice questions where the answer will be supplied and you have to supply the answer. The quiz 00B will be timed. You have 13 minutes to finish this quiz. Normally, in a quiz, you can only take the quiz one time. But since this is the very first extra credit quiz. I'm allowing you 2 attempts on this quiz, but it does have to be finished. Where is it? Finished by January 8th at, oh, excuse me, that's when it starts. January 11th, has to be finished by January 11th at 8 p.m.
Professor, I have a question. I was wondering whether you're going to be making the slides available to us on Canvas or no.
I will after I'm done with the lesson. I'm not finished with lesson 1 yet, meaning chapter 1, chapter 10. When I finish with it, I will make it available. And if I haven't made it available to you, you guys should send me a reminder. Can you make the slides for lesson 1 available? But I'll try and get that done when we are finished. And I hope to finish that, what do you call it, PowerPoint presentation today.
Okay, thank you.
All right. The practice quiz 00B is time. Once you start it, you do have 13 minutes to finish it. Normally, when you take a quiz, you'll only get 1 attempt, but on this one, you get 2 attempts. This one will be graded. I think it's 0.1 point for every correct answer. And then on quiz 00A, it'll be worth 1 extra credit point. Any question about the practice quizzes? You should do practice quiz 00B because it will require you to download Respondus Lockdown Browser and get it running on your system. And I would like you all to get it running on your system for the practice quiz, because if you do have trouble taking it on quiz 1, you will have to contact the computer help desk, and then they'll have to work with you to get it up and running on your system. And you only have a short amount of time to get it up and running on your system before the deadline will expire for the quiz 1B. Okay, so try and get Respondus Lockdown Browser up and running on your system now, and with practice quiz 00B. This is a practice quiz, so I'm not going to be worried about somebody taking this one late. But on the other quizzes, you cannot take it more than 2 days late, because the other quizzes will show answers 2 days after the due date. And after that point, you will not be allowed to take the quiz. Any question about any of that? All right. So let's close that and see. Oh, I need to talk a little bit about the extra credit study. You can form a study group, preferably with somebody in the class, and you need to study it. It gives you the directions in here. Form a study group. You have to study for at least a half an hour per session. For each session, submit a document in the extra credit folder. That's this folder here. Although you don't have to submit a document, you can actually type it in this assignment. Include your full name and the full names of everyone in your group. Tell me when you studied, the date, what you studied, and for how long you studied. You may not study this week's lab, and you will receive a penalty if you do. You are supposed to do your lab work during the lab time. It is not for extra credit studying. During the studying, you should be ideally quizzing each other. I won't require that, but you should be studying something that you will be quizzed on. If everything is submitted correctly, you will get 1 extra credit point for everyone in the group, meaning only one person has to submit it for your group. You can do this for a total of 9 times to get 9 extra credit points, but you must do it once per week. And there is a deadline. I think the deadline for submitting is 11:59 p.m. this Saturday. And you could start, I don't know if that's 12 midnight or 12 noon on Sunday. Anyone remember it's 12 a.m.? I think that's midnight. The easiest way to do that is to just get together over Zoom, but you could get together over your phones. And you can, if you're on campus or wherever, you can get together in person. Okay. When we had class on campus, the students tended to get together on campus, but you don't have to do that. You can use Zoom, you can get to use your phone. All right, any question about any of that? I think that's it. There will be a lecture assignment and then lab assignments due this week, but they're not due until Saturday. And let me remind you, if you didn't get 100% on lab 00 or lab 01, you have until Saturday to turn in corrections. And these corrections for the lab will be for full credit. All right, I think that's it.
Is that a one-time thing?
No, for the lab, you'll always get 2 chances.
Okay, thank you.
For the lecture, you only get 1 chance. And then for all other assignments, you only get 1 chance. But for the lab worksheets, you get 2 chances. The lab, I consider it a learning experience. Let's come on up here. Sorry. All right, let's just go to the lessons. Is this the last slide we had talked about? Yeah, 2 here. So we talked about what comes under the umbrella of microorganisms, and it does include some things that were not cells. Did we talk about this slide? All right, I've got this marked to here, so I'm going to assume we did talk about acellular microbes. All right, there are 3 domains. The slide is reviewing that: bacteria, archaea, eukaryotes. The bacteria and the archaea have the cell type prokaryotes, and the eukaryotes have eukaryotic cell types. The chromosomes differ between the different domains. The eukaryotes have many chromosomes. The bacteria and the archaea typically have only 1 chromosome. And those chromosomes are circular, meaning they form a circle. The starting point is the same as the ending point. That's different from the eukaryotes that have many chromosomes, or typically have many chromosomes, and they're linear chromosomes, like on the X chromosome. I don't know if you can see my fingers. I'm drawing an X. Maybe I'll do it here. It has a beginning point and an ending point, which differ on the linear chromosomes. They also differ that the bacteria have no histones on the DNA of the chromosome, and the eukaryotes do have histones. This is a protein that is associated with the DNA of the chromosomes. We'll talk about the introns later in the term. Bacteria have no introns. Eukaryotes do have introns. And the archaea are slightly in between. Sometimes they're like the characteristic of the bacteria, and other times characteristics of the eukaryotes. The ribosomes differ between the domains. The bacteria and the archaea have small ribosomes that we call 70S in size. The S is the sedimentation coefficient, where the ribosome sediments on an ultracentrifuge gradient. It's just how the biochemists measure ribosomes. And do you need to know that? No, but you do need to know that the ribosomes are 70S in size. The eukaryotes have a slightly larger or somewhat larger ribosome, and we call it an 80S. So it sediments deeper down on the ultracentrifuge gradient. The cell walls differ. The bacteria have peptidoglycan as the main molecule in the cell wall of bacteria. The archaea have no peptidoglycan. The eukaryotes have no peptidoglycan in their cell wall or in their cells. And of course, animal cells do not have cell walls. Any question about any of that? If not, let's talk about a brief history of microbiology. It largely concerns the debate over whether spontaneous generation could occur or whether the theory of biogenesis accounted for life. And we'll talk about that. It also, and when we talk about the brief history of microbiology, covers the golden age of microbiology, when microbiology was first starting from 1857 to 1914. In these slides, you do not generally need to know the dates. In the golden age, it's just whenever people looked at something under the microscope, they generally were seeing in the golden age something brand new, and it was a new discovery. Any question about any of that? And that's a short history of microbiology with the main events, or I should say a number of the main events, with the golden age being in gold here. Now, whenever I give you a slide on the history of microbiology, you do not need to know the dates. I'm going to just state the dates because that's one way to talk about the events, but you do need to know this first one, and that is slightly greater than 3.5 billion years ago. The ancestors of bacteria were the first life seen in the fossil record on Earth. And here we're actually seeing it here. That's the actual fossil, and there's an artist's rendition of it. And then there's other fossils there. And these were all bacteria, or I should say the ancestors of bacteria, because back then life wasn't really bacteria. It was a prokaryotic cell, which really was the ancestors of all life, but we're saying the ancestors of bacteria. And then the next important event was the Babylonians wrote down on their clay tablets recipes for making beer. You don't need to know the date, but that's 4,300 BC. They wrote these down. And the reason why this is important is this is a recipe using microorganisms to make beer. Now, the Babylonians didn't know anything about microbes, but they did write recipes for making beer. And it's the first record we have for the use of microbes. And then the next one at 600 AD, the Mayans wrote records for making a fermented beverage, cacao, which we would call chocolate. The next important event in microbiology was Janssen developed the compound microscope. You could not see microbes until the invention of the microscope. And you don't need to know the year, but it was in 1590. Surprisingly, nobody took the microscope to look at living things until 1665, when Robert Hooke used a microscope to look at living things, and he saw little boxes that he called cells. Let me see if I can do a slight drawing to illustrate this. Having trouble remembering how to get that. I just want paint. I don't remember how to get paint on this computer. All right, let me stop the sharing and I will get paint by opening up a picture. Come on. Hope I didn't move that. Yep, I did. Darn it. Ah! See if that undoes it. Nope. Yeah, I moved it back. All right. Go to pictures. Open one up. There we go. Now share my screen. That's really a lot of work, but a student once complained about a picture I opened up, which was just a diver, and it caused all sorts of problems. I'm not ever going to do that again. So, all right. When Robert Hooke looked at, why isn't that writing? Looked at the first cells under the microscope, he saw that they were shaped something like this. I'm not doing a real good job drawing. I'm not getting the arrow all the way up. There, that's better. And this reminded him of the cells in a monastery, which the monks, they'd have their hallway here, and then a door here. And this is where the monks lived or slept and did their monk things. And it reminded him of the cells in a monastery, so he called them cells. And that's where the term comes from. Any question about any of that? Go back to my PowerPoint. That marked the beginning of the cell theory, which is that all living things are composed of cells. This was developed further and actually written up as the cell theory by Virchow many years later. The above events I talked to you are not specifically microbiology, like Jensen developing the compound microscope. However, these things needed to be known before microbiology could begin. And like I said, Robert Hooke was the first person who at least wrote about it, used the microscope to look at living things, and he saw cells. Now, he looked at cells in plants, and plants initially were seen. When they looked at animal tissue, they did not see cells because the microscopes were not good enough. They were too blurry that people could not see the cells for almost 100 years after 1665. And why do you think the cells of plants were much easier to see than animal cells? Anyone have a guess? Nobody has a guess? Well, it's simply that plant cells have cell walls, and that made the boundaries very easy to see, whereas animal cells do not have cell walls, and they had to wait until the microscopes improved, especially their, their focusing and their non-blurriness, and then they were able to see cells in animal cells that lack the cell walls. All right, the early history of microbiology concerned two hypotheses about how life originated. The two hypotheses, and you could call them theories, were spontaneous generation, that life arises spontaneously out of inorganic matter. A vital force, the theory was, comes in to the inorganic matter and does something to it, and then life spontaneously generates. The competing hypothesis was biogenesis, and that is life only arises from other pre-existing life. Like everybody knows that a baby comes from two parents, and they didn't ever argue a baby human came generated spontaneously. But there were other statements about the debate of spontaneous generation. Like over a century ago, people stated that when you throw in a log into the fire, sometime a newt arises out of the fire. And they said spontaneous generation, the wood and the fire spontaneously generates a newt. And then people saw flies emerging from manure and they said, spontaneously generation. The vital force got into the manure and then the flies emerged. And then maggots arising and decaying corpses is another example that was used as spontaneous generation. Most were logical, but not scientific. Some were amusing. Somebody wrote that if you put sweaty underwear and wheat in a big jar, it would spontaneously give rise to mice. In reality, what happened was the mice smelled or discovered the wheat and then got into the jar to eat the wheat, and then they got trapped in the jar and couldn't get out. And so the sweaty underwear had nothing to do with it, actually. And then, of course, all the others had an explanation too. The newt was inside the log when it was thrown into the fire, and the newt noticed the heat, and so it quickly emerged to try and save itself. Let's skip that. The first evidence against or for spontaneous generation was done by Francesco Redi. And actually, you should know this not only for the first evidence against spontaneous generation, but he also is the first person to document and write up a controlled scientific experiment where he changed the variables and then wrote it up. What he did was he took 3 jars that he completely sealed, and then 3 open jars, and he put meat inside the jar before sealing them, and he saw no maggots. And in the open jar, he saw maggots. And the spontaneous generation people said, no, by sealing the jar, you prevented the vital force from getting in, so that's why there were no maggots. So what he did was he took 3 covered jars and covered them with a fine wire net. And then he had 3 open jars and 3 completely sealed jars. And the jar with the wire net got no maggots. The 3 open jar got maggots. So the results did not support spontaneous generation. Okay? Any question about any of that? After Francesco Redi's time, everyone agreed that fairly complex organisms could not spontaneously generate. However, microbes, they said, could spontaneously generate. And the reason was they would have sterile broth, and then they might even seal the tube, and then it would become cloudy with microbial growth. And we call that turbid. Okay, any question about that? And this was a little more difficult to disprove spontaneously generation there. But what happened was, let me hold off on that and state, about this time marked the beginning of microbiology, where somebody finally looked at microbes. And this was done by Antony van Leeuwenhoek, the best microscope builder of his day. And not only did he make microscopes by hand himself, he actually ground his own lenses. And he was the best lens grinder of his day. He may have had also extraordinarily good eyesight, and he used his own microscope to look at a bunch of things like microorganisms in scrapings from his teeth, and microorganisms in rainwater and peppercorn infusions. And he looked at the water from the River Thames, which people had been drinking out of in the city of London. And he found microbes everywhere he looked. Not only did he see the first microbes, but he actually saw the first bacteria, cells so small that they were not seen again for nearly 100 years. And the reason why he saw them was because he had great microscopes and everybody else had poor microscopes and they could not see bacteria. But if we look at his drawings here, that looks like a bacilli, that's a bacilli, and he's showing its movement. This looks like a sporillum. These look here like cocci, and those are more bacillus. Drawings of actual bacteria that Antony van Leeuwenhoek drew. And it took 100 years for people to improve the microscope before anyone else could see bacteria. All right, any question about that? So Antony van Leeuwenhoek is frequently called the father of microbiology or bacteriology because he's the first one who actually looked at microbes. All right. Around this time, Rudolf Virchow proposed the cell theory, meaning people had the idea of the cell theory that life only exists from pre-existing life, meaning from other living cells. And this became known as the theory of biogenesis. But Rudolf Virchow was the first to write it up in the scientific literature. And this started, once again, the debate of whether spontaneous generation could happen among microorganisms or whether the theory of biogenesis occurred, meaning that microbes could only occur from pre-existing microbes. And that debate was settled by Louis Pasteur, who disproved spontaneous generation and therefore confirmed the theory of biogenesis. He demonstrated that microbes are present in the air and that microbes could be destroyed by heat. He did famous swan neck flask experiments where he took a glass flask like this, so it has a long glass neck, and he put the media in, and then he heated the glass and bent it, and they call that a swan neck. And then he heated the media, sterilizing it, and then he left it open to the air so the vital forces could come in to the media and change it to life, and he saw no microbes. What he noticed was you don't get any microbes here unless you tilt the media and put the media down here and then bring it back. And what's happening is the microbes from the air could come into the media, contaminating it, and then grow microbes in the media. But in the swan neck, the microbes from the air would come in, but then by gravity, they'd settle here. And they would not come up gravity and then get into the media. Any question about any of that? So this experiment disproved spontaneous generation for microbes. And I can tell you, if you ever go into the microbiology lab, a professor set up one of these open in the microbiology lab. And the last I checked, and that was probably a couple of years ago, it was over 10 years old. So now it's probably over 15 years old. And there are no microbes growing in the media. Eventually, the media is going to all evaporate, in which case we'll have to start over again, I guess. All right. So the golden age of microbiology was the early times of microbiology. Generally, the beginning with Pasteur's work, which showed, in a sense, he didn't show, but he gave rise to the idea that there's a relationship with microbes and disease. This became known as the germ theory of disease. And out of Pasteur's work came the idea of immunity. And then out of all of this came the concept of antimicrobial drugs, that we could find a drug to use against microbes and then prevent or cure disease. So in 1850, you don't need to know the date, but Pasteur developed the basic idea of aseptic technique. And that's extremely important because you need aseptic technique to have a pure culture. And without studying a pure culture, it's really difficult to know what's going on. Like if you have two microorganisms in a tube and one of them ferments the media, you don't know which one did or if both did. And so you need to work with a pure culture in order to study it. Shortly at this time, Koch developed a technique for streaking for isolation. This was really important because it made an easy way to get a pure culture. And that is, you just use a solid media, streak on it, and then from your colony, it's likely derived from a pure culture. And if not, and actually it's only about 75% pure, you can streak it out from that one colony onto another plate to get other colonies. And if you send it through 2 colony isolation events, you'll get a pure culture greater than 95% of the time. Any question about that? About the same time, somebody working in Dr. Koch's lab, meaning one of his technicians, developed nutrient agar, a new and cheap solid medium for growing mini microbes. And also at this time, Sergei Winogradsky developed the idea that microorganisms are involved in the biogeochemical cycles, like the nitrogen cycle. It's curious that Sergei Winogradsky studied microbes in a, what do you call that? An ecosystem where there are many different microbes, and he had to do that to get the biogeochemical cycles. Most of the other microbiologists in his time studied pure cultures, but Sergei Winogradsky could not have studied the biogeochemical cycles if he only used pure cultures. Louis Pasteur also discovered the concept of fermentation, that alcoholic fermentation occurs from the conversion of sugar to make alcohol, which is used in the making of beer and wine. And he also discovered that microbial growth was responsible for spoiling some food. Like when beer or wine became sour, it was because there's another microbe in the beer or the wine growing and converting the alcohol into acetic acid, the vinegar, and that would spoil the beer or the wine. Louis Pasteur then invented pasteurization, the application of high heat for a short period of time, which is used in a variety of foods to help preserve them, like pasteurized beer. And that's exactly what Louis Pasteur developed pasteurization to do, to pasteurize beer to prevent it from spoiling. And what he did was he used a heat that wasn't hot enough for long enough to evaporate all of the alcohol in the beer or the wine, but it did heat it to prevent the growth of the spoilage microorganisms. With the concept of fermentation occurring with microbes, people then ask the question, do microbes have a similar relationship with plants and animals? And might microorganisms cause disease? This concept became known as the germ theory of disease. This was important because originally people believed disease was caused by poisonous vapors that were breathed in, causing diseases. Like, for example, they thought cholera was obtained from breathing the poisonous vapors when somebody would go walking through a swamp. Any question about that? In reality, what happened was the person walking through a swamp got exposed to cholera and then perhaps breathed it in and then got the disease from cholera. It wasn't because of the poisonous vapors. I'll let you read about this. The germ theory of disease was actually kind of proven by Schamal Weiss, and I could be butchering that name. I'm not German or Austrian, and Lister, who actually showed that if you simply wash your hands or use a chemical disinfectant, you could reduce the chance that a hospital patient could get an infectious disease, which really proved the germ theory of disease. Well, not really proved. What does that help? That's evidence for, I guess. When you wash your hands, there's less likely a chance that the patient would be getting an infectious disease. Robert Koch literally proved it by showing that the bacterium Bacillus anthracis causes the disease anthrax. And he provided the experimental steps he used to prove that that specific microbe, Bacillus anthracis, causes a specific disease, anthrax. These experimental steps are known as Koch's postulates, and Robert Koch wrote all this up. In 1876, he wrote up that Bacillus anthracis causes anthrax, and then he wrote up Koch's postulates in 1884. You don't need to know the date, but this proved that the germ theory of disease was correct because an microbe caused disease. Around this time, we also had the first vaccination, Edward Jenner in 1796. You don't need to know the year. Showed that if you inoculate a person with the cowpox virus, that person was protected against smallpox. And this vaccine was highly used at the time because nobody wanted to get smallpox. And it was not understood how his vaccine was protecting against smallpox. So it was not for 100 years that the next vaccine, or around 100 years, was ever developed. And once again, it was Louis Pasteur who showed what was going on and why a vaccine is protecting an individual against a disease. What Louis Pasteur showed is that the cowpox virus was genetically similar enough to smallpox. And so when the immune system mounts a response against cowpox, it also mounts a response against smallpox, giving the person immunity to smallpox. I got a message stating that my internet is unstable. So if my signal is breaking, just let me know. And if we do get disconnected, I will try to reestablish the connection. So just ask me to repeat if something isn't clear. The term vaccination is actually derived from vaca, which is a word which means cow. So Vaccination comes directly from Jenner's cowpox virus being used to protect against smallpox. And this protection is called immunity. Any question about any of that? All of this work actually gave rise to the concept of chemotherapy, the treatment of disease with different chemicals. Chemotherapeutic agents can be used against infectious disease. We don't usually call that chemotherapy because chemotherapy is today used specifically to mean an anti-cancer treatment, meaning a chemotherapeutic agent is usually a anti-cancer drug. But widely speaking, you can say a chemotherapeutic agent can be used against any disease. We do tend to use antibiotics or an antibacterial agent to mean a drug that specifically works on an infectious disease caused by a bacteria. You can have synthetic drugs, meaning man-made, or drugs which are made by a living organism. Antibiotics, by the textbook definition, is a chemical that is produced by a living organism, usually a bacteria, an archaeobacteria, or a microbe that's a eukaryote, like a fungus, that inhibits or kills other microbes. Now, that is the textbook definition. Most clinicians and most patients, most people, do not make this distinction, meaning penicillin G is made by a living organism, so it is, by the textbook, considered an antibiotic. And penicillin G and carboxycillin is a synthetic man-made drug, hopefully I got the right one, which is, by the textbook, not an antibiotic. It would be a synthetic drug, okay? Most clinicians and most people do not make that distinction, but a textbook does. The first chemotherapeutic agent used against an infectious drug was actually quinine from the bark of the cinchona tree, and it was used to treat malaria. Paul Ehrlich used all of this to come up with the concept of a magic bullet, which he actually found one in 1910. Once again, you don't need to know the year, where he found a drug that could be used to treat syphilis. And he called that the magic bullet because it could kill the disease-causing organism, but not kill the patient. Any question about any of that? So Ehrlich came up with the first drug to cure the first infectious disease other than quinine, which was one naturally discovered. You're just gonna have to remember all these things. And I'll talk more about the discovery of the antibacterial drug later in the term. But in 1910, Paul Ehrlich came up with the first magic bullet, coming up with a drug which could cure syphilis. And from that day on, people were looking at different chemicals which could be used as a antimicrobial drug. Alexander Fleming discovered the first antibiotic in 1928. What he did was he streaked out a plate of bacteria, and he was sort of sloppy and got contamination on the plate. And then he just left the plate on the counter where both the bacteria and the fungi, which was the contaminant, could grow. And then he came back over the weekend and saw the plate on the counter and noticed that around the mold, there was a region where there was no bacteria growing, although he streaked there. And he correctly concluded that something was coming out of the mold, preventing the growth of the bacteria. And this we call an antibiotic. So penicillin was the first antibiotic that was discovered. However, it was not the first antibiotic that became clinically available. And it just was difficult to purify penicillin G from the media that the mold was growing in. And in 1928, we just didn't have the technology to do that. And Alexander Fleming wasn't somebody who could purify a chemical anyways, but all he could do was have a cell-free extract of the media. The first clinically available drug was sulfonamide, which is a sulfa drug, and it was actually man-made, and so they could make it pure. And it became available in the clinic as early as 1935. Penicillin became the second clinically available, at least family of drugs. There might have been another sulfa drug that was developed before 1940, but penicillin became clinically available and mass-produced in the 1940s. All right, bacteria were once classified as plants, and that's why we use the term flora to describe microbes. If you ever hear me use the word microflora, that's because I was trained in microbiology long before that was, well, that's what microorganisms were called, part of the microflora. And it's not now used, it's now replaced by microbiota, and I'll try and use that. But if ever I make a mistake and use the word microflora or flora to describe microbes, you can just correct me and say that term is no longer used anymore, and I'll say thank you. It's now microbiota is the term we use to refer to microbes, especially microbes that are growing on or in a person, animal, or plant. And we call that the normal microbiota, the microbes that are normally present in and on animals and plants. For the human normal microbiota, that would be the normal microbiota that are found in and on a human. And they do differ for different species, like Bartella henselae is a bacteria that is the normal microbiota of cats, but it's not normally found on humans. So it is not part of the normal microbiota of humans. Any question of that? So animals and plants host a symbiotic microbial community, their normal microbiota, which are many species living together in a community. And these microbes are central to their welfare. The normal microbiota are found on and in this, well, not in the skin, but on the skin, in the oral and nasal cavities, in the respiratory tract, usually not the very low regions of the respiratory tract. You would call it pneumonia if you get in the lungs, for example. But it's found in the upper respiratory tract. It's found throughout the digestive tract, in the urethra. It's usually the normal microbiota are not usually found internal, such as in the blood, the internal organs and tissues. The normal microbiota are important in preventing the growth of various pathogens. They can compete for resources and prevent the pathogen from growing or getting established. They can secrete toxins that can inhibit the growth of the pathogen. We'll talk a little bit more about that later. They can produce nutrients and growth factors for their host. For humans, for example, part of our normal microbiota make a significant amount of our folic acid and vitamin K. So they can aid the welfare or the host by competing against the pathogen, and then providing nutrients and growth factors for the host, as well as helping the host digest their food. If you don't know, we have tons of microbes in our gut, like E. coli, which helps us digest our food. The normal microbiota do not normally cause disease in a normal, healthy host. However, they can cause disease in certain circumstances. Before I talk a little bit, well, let's go ahead and back up down there. For example, AIDS patients don't have an immune system, and so the normal microbiota can cause disease in an AIDS patient. Any question about any of that? All right. So infectious diseases are diseases that are caused by a microbe that causes the disease, and we call that microbe a pathogenic microbe because it can cause disease. And the pathogen has to overcome the host resistance before you'll get disease. For example, if you catch actually most of you are young, so even if you catch COVID-19, most of you will be asymptomatic. But if you catch the cold virus, and particularly if you get lots of sleep, you may not ever have any signs or symptoms of the human cold. And what's happening is the infectious disease is in you, but it is not overcoming the host resistance. And so it is not causing at least signs or symptoms of disease. So in a sense, it's asymptomatic. And actually, most of you are young, so COVID-19, for example, can be asymptomatic in you guys. Any question about any of that? All right, so that's an infectious disease. Let's talk about an emerging infectious disease. This is a new or changing disease that is increasing or has the potential to increase in incidence. A current emerging infectious disease is COVID-19 because I don't remember, more than 3 years ago, something like that, this disease was not in the human population. So it is a current emerging and infectious disease. A bird flu is one that they are worried may become a disease which will increase in incidence, in which case it is an emerging infectious disease simply because sometime in the future it has the potential to increase. Any question about any of that? All right. The remainder of the slides talk about emerging infectious diseases. I'm not going to test you on this other than what an emerging infectious disease is. So I'll let you go over the other emerging infectious diseases if you care to do so. I'm not going to take the time to do that. All right. Any questions about chapter 1 and chapter 10? If not, let's end that chapter. and begin chapter 2. How is everyone doing? Just want to
Good.
check in. Okay, good. Just want to check in, see everyone's still there. All right. Chapter 2, chemical principles. As always, I'll give you a slide listing the major goals and a rough outline of what we were covering. Let me remind you that this is not all of the goals. While I'm seeing that, I'll publish chapter 1 and chapter 10 now. And for all of the goals, you want to look at the objectives for chapter 2. These are only the major objectives. Know the terms. What's an organic and an inorganic compound? Know what a hydrocarbon is. What's a polar molecule, a non-polar molecule? Know what hydrogen bonds are, acid, base, and pH. You should also know, with respect to the 4 major biological molecules, be able to describe their relationships and characteristics, including what's a monomer of that biological molecule and what's a polymer. Be able to describe the composition, the structure, and function of these 4 major biological molecules, and be able to give examples of each. What are the 4 major biological molecules? You can actually see 3 of them on any box of cereal or any ingredient listing what's in the something bag that you're eating. Okay, you can find 3 of them on any box of cereal, for example. What are the 4 biological molecules?
There's proteins, lipids, carbohydrates, and nucleic acids.
Yeah, you got it. Those are the 4 biological molecules. We call them that because they are only seen in something that is alive or was once alive. Like when we look at a rock on the planet Earth and there's a fossil in it, that would be containing a biological molecule that was once alive. You do not find any of the biological molecules when you look at the rocks from the moon, for example. All right, in chapter 2, you should know the scale of things. Let me see if I can blow this up. Come on. This is in the way. So you should know that humans are larger than almost anything else we'll look at. Eggs are smaller than humans. That's a chicken egg. Frog eggs, that's about the smallest thing we can see with our eyes unaided. Smaller than a chicken egg. And then we have eukaryotic cells. Bacteria, smaller still. Viruses, smaller still. Large molecules, smaller molecules like water. A protein would be a large molecule. And then smaller than a small molecule, we have atoms. You do not need to know the size of things. You probably should know that bacteria and eukaryotic cells, typically you need a light microscope to see them. Viruses, though, you cannot see, or large molecules, you cannot see with a light microscope. You'd have to see them with an electron microscope. You should also know that in this measurement here, there's a mistake right there. That's not a micrometer. That should be a a millimeter right there. And it's surprising this was never corrected because this slide had been used for many iterations of the book. All right. Any questions about any of that? You should know that what an organic compound is compared to an inorganic compound. An organic compound is typically complex structurally. I'll show you some of those, and they're just a complex molecule. They have carbon, which tends to hold the molecule together, and carbon can bind to 4 other atoms. Why carbon is sort of the backbone of the organic chemical. Organic chemical compounds are held together typically mostly by covalent chemical bonds. They can have hydrogen bonds in them, but typically the covalent bonds hold the molecule together. Inorganic compounds typically lack carbon. They don't always, but they typically do lack carbon, and they're typically much simpler than an organic compound. Inorganic compounds are required by living organisms for their growth, for their repair, for their maintenance, and their reproduction. Some examples of an inorganic compound include oxygen, water, and carbon dioxide. Carbon dioxide is an inorganic compound, but it does include carbon. Okay? And I did say typically they lack carbon, but they don't always lack carbon. Water is one of the most important and most abundant molecules for life. And so we'll talk about water in our next couple of slides. Any question about any of this? Let me just show you what I mean by complex molecule. Got to go down several slides. There we go. That is a fairly complex molecule, and it's an organic compound. These two, we'll talk about them later, are fairly complex molecules. The simplest organic compound is this molecule here. Well, I guess, or that molecule there. And like I said, those are the simplest ones. Most of them are more complex, like this molecule. All right, let's talk about water. Water is a polar molecule. A polar molecule is one that has a partial positive charge on one end of the molecule and a partial negative charge on the other end of the molecule. And let me warn you that your textbook author doesn't describe it as a partial charge. I don't remember the term that he uses. Oh, he calls it a slight charge. And I think partial charge is a better term. A partial charge is not a complete charge. If it was a complete charge, we'd have a +1 on the hydrogen, in which case it would be lacking 1 electron to be a +1. And on the oxygen, if it was a -1, it would be adding a complete electron to the oxygen. Okay. And with water, it's a polar molecule. It has a partial charge with the hydrogen and a partial negative charge with the oxygen. And why it has this partial charge, do I discuss that here? I don't know where I discussed that. What happens is that the oxygen pulls harder on the electrons of this chemical bond. So these electrons spend more time with the oxygen than they do with hydrogen. But the electrons do spend some time with the hydrogen. And that's why we have a partial charge difference. And we call this a polar molecule. And just for your information, this hydrogen also has a partial positive charge. The polarity of water can cause hydrogen bonds between different water molecules. Let me blow this slide up. Do that. There we go. That's what I needed to do. Come on. There we go. Move that up. Yeah, let's go there. So water is a polar molecule having a negative charge in the oxygen, a positive in the hydrogen. That can create a hydrogen bond between different water molecules, where this positive hydrogen will be electromagnetically attracted to the negative of this oxygen. And that attraction we call a hydrogen bond. Any question there? This water molecule, as you can see, can actually form 3 or 4 hydrogen bonds, one with this water molecule, one with that water molecule, and then 1 or 2 with these water molecules. So water has 2 chemical bonds. It has a covalent bond within the molecule between the oxygen and the hydrogens, a chemical covalent bond, and it is a polar covalent bond. And then between water molecules, we have hydrogen bonds. Between this water molecule and that water molecule, we have what do you call that? A hydrogen bond. So remember, water has 2 types of chemical bonds. Water is a good solvent. All polar molecules and ions can dissolve in water. The reason being that the positive charge of that molecule, which is this little green one here, water can come in, and then water will surround the positive charge, putting the partial negative oxygen towards the positive charge. And then the waters will surround the positive charge like that, so that ionic molecules can dissolve in water, as well as polar molecules can dissolve in water. Any question about that? And for the chloride ion, which has a negative charge, it'd be surrounded by the water with the hydrogens oriented towards the negative chloride ion. So why water is a good solvent? Just because it can dissolve all polar molecules and ionic molecules, which are a lot of molecules. It's not all molecules. For example, water cannot dissolve oil or a fat, and that's because the oil is not polar. So water is a good solvent. Polar molecules are hydrophilic, and that means literally fear of water, but we don't really mean fear of water. It means they do not dissolve in water, and that's because they will stay away from water. When you put oil and water together, oil is a non-polar molecule. Water is a polar molecule, and then when you mix them, they will separate. Any question about any of that? So oil is non-polar. That means it's hydrophobic. It literally fears water. It will not dissolve in it. Okay. An example of a hydrophilic molecule is actually acetic acid, which is part of vinegar, or anything that can dissolve in water, such as sugar. It's hydrophilic in a polar molecule. Any question about that? Did I say that wrong? Hydrophilic actually means water lover. Hydrophobic, water, fear of water. Any question about any of that? All right, if not, I think I'll stop here because it's time to end the lesson, and I'll see you at 6:30 for the lab. Any questions? All right, I see you at 6:30.